So imagine you are standing in the middle of a grocery store aisle, and in your left hand, you are holding a bottle of pure glucose water.
Okay.
And in your right hand, you have a beverage sweetened entirely with pure fructose. Now, if you look at the nutrition labels, they contain the exact same number of calories.
Right. The exact same amount of chemical energy.
Exactly. So the big question is, if you drink them, will your brain treat them as identical fuel sources? Because for decades, science confidently told us, yes, a calorie is a calorie.
Which makes intuitive sense, right? But it's totally wrong.
It is. And that brings us to the mission of our deep dive today. A new paradigm-shifting paper just dropped in the journal Neuron by McKnight and colleagues, and it completely shatters that old idea.
It really does. It fundamentally changes how we view nutrition and, honestly, how we understand cravings.
Yeah, so today we are going to rewire your understanding of hunger.
Yeah.
We're going to break down this newly discovered hidden gut-brain highway and explain exactly why things like high-fructose corn syrup have such a vice-like grip on our diets.
Because what this research team did was basically map the microscopic mechanisms of hunger. I mean, we always assumed the brain's hunger centers were just like blind bean counters.
Right, just tallying up total calories hitting the stomach.
Exactly. But this paper proves that the gut is actively identifying the specific molecular makeup of what you eat. And it routes that information to the brain through entirely separate cellular networks, just based on the type of sugar.
It's wild. So to really visualize this for everyone listening, we need to look deep inside the base of your brain. There's a region called the hypothalamus.
Right.
And I want you to picture a cluster of cells there called AgRP neurons. That stands for agouti-related protein.
It's a mouthful, yeah.
It is. But just think of this cluster as your body's master survival switchboard.
Mm-hmm.
When those AgRP neurons are firing rapidly, you feel a deep, nagging, ravenous hunger. Like you will do almost anything to find food.
Yeah, you are biologically driven to eat.
Right. And the traditional thinking was always that when food finally hits your gut, these neurons just naturally power down, telling your brain that you're satisfied.
This is true, but we can actually watch this happen in real time now, which is where the study gets truly fascinating.
Yeah, tell us about the setup they used.
So the researchers used this mind-blowing technique called fiber photometry.
Oh.
They literally injected a harmless, genetically modified virus that carries a calcium indicator. It's called GCAM MP6s.
Wait, they injected a virus directly into the brains of mice?
Exactly, right into the hypothalamus. And neurons use calcium to fire electrical signals. So this virus alters those AgRP neurons so that whenever calcium rushes into the cell literally glows with a fluorescent green light.
Wow. So you are basically installing a microscopic flashbulb right on the hunger switchboard.
That's a great way to picture it. You can literally see when the mouse is hungry because its brain is glowing green.
That is incredible.
Yeah. So they took fasting mice whose hunger neurons were glowing brightly because, they were starving, and they gave them an 8.3% glucose solution to drink.
And what happened?
Almost instantly, the fluorescent glow dropped. The AgRP neurons rapidly powered down. The brain registered the energy and shut off the hunger alarm.
Which makes sense. That's the textbook response.
Right. But then they took a different group of fasting mice and gave them an equicaloric 8.3% fructose solution. Same volume, same exact number of calories.
And here's the crazy part. The neural activity barely dimmed.
Barely touched it.
The flashbulb stayed lit. The hunger switchboard just stayed completely on, as if the calories hadn't even arrived in the stomach.
Which is so bizarre. And, to rule out the possibility that glucose just tastes better to a mouse, the scientists surgically bypassed the mouth entirely.
Right, because maybe the tongue was doing the work.
Exactly. So they implanted intragastric catheters right into the stomach and intraduodenal catheters straight into the upper intestine.
Wow.
Yeah, they infused the sugars directly into the gut while the mouse was totally unaware, no taste receptors involved at all.
And the result held perfectly, right?
Perfectly. Glucose sharply crashed the hunger signal, and fructose barely touched it. They even tested a 50-50 blend of glucose and fructose, which is just common table sugar, sucrose.
Right.
And that resulted in a perfectly middle ground neural response.
So it's almost like having a highly exclusive VIP club. Let's use an analogy here.
Love a good analogy.
So if your AgRP neurons are the bouncers working the door of the hunger club, glucose walks up, flashes a VIP pass, and the bouncers immediately shut the club down and send everyone home.
Right.
But fructose just sneaks in through the back alley, The bouncers never even see it, so the hunger party just keeps raging on?
That is exactly what's happening at the cellular level.
But wait, hold on. I have to push back here. If fructose is sneaking past the bouncers and the hunger switch is still completely on end, shouldn't those mice be absolutely tearing into their food right after? Like, why aren't they ravenous?
See, that logical leap is exactly what makes the biology so beautifully complex. And honestly, the researchers assumed the exact same thing.
Oh, really?
Yeah, so they set up a clinical-style satiation study. They preloaded fasting mice with either a gastric infusion of fructose or glucose. They waited a bit and then gave the mice free access to normal chow to see who would eat more.
Right, because if the fructose mice have active hunger neurons, they should eat way more.
Exactly. But they ate the exact same amount of chow.
Which presents a massive biological paradox. I mean, how do they stop eating if their central hunger neurons are still glowing green and firing?
So to explain this, let's do a quick recap of what we've covered so far, just to cement this in your memory before we solve the paradox.
Perfect, yeah. So first, AgRP neurons track specific nutrient identities, not just blind caloric values. Second, glucose deeply inhibits those hunger neurons, while equicaloric fructose only weakly inhibits them.
Right. And third, this all happens post-ingestively, deep in the gut, completely independent of taste on the tongue.
Exactly. So next time you drink a heavily fructose-sweetened juice versus, say, eating a starchy snack that breaks down into glucose, I want you to pay attention to your body. Ask yourself, does your deep, nagging sense of hunger fade differently?
It's a great question to ask yourself, because the answer to our paradox, why they still stop eating, lies in physics. Specifically, something called gastric distension, or the balloon effect.
Okay, tell us about the balloon effect.
So fructose has a unique physical property compared to glucose. It is highly osmotic.
Meaning it interacts with water differently.
Exactly. When it hits the intestines, it draws a massive amount of water out of the surrounding tissue and pulls it right into the gut. It physically stretches the walls of the intestines significantly more than glucose does.
Oh, so it's literally inflating the gut like a water balloon.
Yes. And that physical stretch acts as a completely separate mechanical stop-eating signal to the brain. It is entirely independent of those nutrient-sensing AgRP neurons we've been talking about.
Wow, so it's just sheer physical pressure.
Right. And to prove this definitively, the researchers used a brilliantly elegant tool called optogenetics. Have you ever heard of this?
I have, but let's break it down for the listeners because it sounds like sci-fi.
It totally does. So
Yeah.
they implanted a tiny fiber optic cable into the mouse's brain.
Into the brain.
Yes. The original virus they used, the one that made the cells glow, also inserted light-sensitive ion channels into the AgRP neurons. So by shining a specific 450 nanometer blue laser directly onto those neurons, the light physically forces the ion channels open.
Wait, so you are manually hijacking the brain with light. You're forcing the hunger switch to stay on, no matter what the stomach is actually saying.
Exactly. It completely overrides all natural signals. So when they forced the AgRP neurons to stay on during a glucose infusion, the mice just kept eating.
Because the glucose relies on those neurons shutting down.
Right. That proves that glucose completely relies on shutting down those central AgRP neurons to make you stop eating. But when they forced the AgRP neurons on during a fructose infusion,
The mice still stopped eating.
yes, because they were physically too full from the water balloon effect.
Oh my gosh. It's like having two totally different fuel gauges in your car.
I like that. Walk me through it.
Okay, so one gauge, the AgRP neurons and the glucose, that measures the actual chemical energy in the tank. But the other gauge, the distension and the fructose, it just measures if the physical metal of the tank is stretching outwards because it's packed with liquid.
That is exactly it. Both gauges tell you to stop pumping gas, but for totally different biological reasons.
And we can see this play out in our own lives, right? The mechanical distension triggers a temporary satiation pathway that simply overrides the central hunger neurons for a short time.
Yeah, it's a very short-term fix.
Which brings up a great question for you to think about. Have you ever eaten a massive meal and felt physically stuffed, maybe even to the point of pain, but your brain still mentally craved dessert?
Oh, I've definitely been there.
Right. You were experiencing that exact disconnect. Your mechanical distension gauge was maxed out, but your neural satiation gauge hadn't registered the right chemical drop.
Exactly. Which leads us to the next big question. If this weak neural response to fructose doesn't actually change how much we eat in the short term, what is the point of it?
Right. What is the evolutionary purpose?
It dictates your long-term food preference. It turns out your AgRP neurons are the master architects of what foods you will choose to eat tomorrow and the day after that.
Oh, wow. So they're actively programming us.
Yes. And to prove this, the researchers used another mind-bending technique called chemogenetics, specifically a system known as DREAD.
Dready.
Yeah, it stands for designer receptors exclusively activated by designer drugs.
Okay, that sounds even more like pure science fiction than the laser thing.
It is incredible engineering. They bred mice to have specialized synthetic receptors called HM4DI located exclusively on their AgRP neurons.
Just on the hunger neurons.
Right. In a normal environment, these receptors do absolutely nothing. But
Yes.
when scientists introduce a specific synthetic designer drug called CNO, it binds to these designer receptors and opens up potassium channels.
Okay, getting a little technical here. Break that down for us.
Sure. So positive ions leak out of the cell, which makes the inside of the cell more negatively charged. In neuroscience, we call this hyperpolarization. It mathematically makes it much harder for the neuron to fire an electrical signal.
So you are basically playing the hunger neurons like a volume dial.
Mm-hmm.
But you're doing it without feeding the animal a single real calorie.
Exactly. It's an artificial dial. And this allowed them to set up a choice experiment to see how neural drops drive behavior.
Okay, what was the setup?
They gave the mice a totally novel flavor, let's say grape Kool-Aid. 5 minutes after the mouse drank it, the researchers injected a low dose of CNO, which artificially created that weak, shallow drop in hunger that fructose normally provides.
Right, a tiny drop in volume.
Yeah. Then the next day, they gave the mouse cherry Kool-Aid and injected a high dose of CNO, mimicking the deep, satisfying crash in hunger that pure glucose provides.
And just to be absolutely clear for you listening, both Kool-Aids were 0 calories.
Yes, very important point.
It was literally just flavored water. The only difference was the artificial chemical dial turning down their hunger neurons. So what happened when they had to choose?
Well, initially, the mice had no preference between cherry and grape. But after a few days of this training, when put in a cage with both bottles, the mice overwhelmingly and aggressively chose the cherry flavor.
Wow.
They literally learned to prefer the sensory flavor that subconsciously turned off their hunger neurons the most effectively.
That is terrifying. Let's look at the macro behavior here, because this is exactly where high-fructose corn syrup, or HFCS, enters the picture.
Oh, absolutely.
HFCS is typically a 55 to 45 split of fructose to glucose, and we are constantly drinking massive 32-ounce sodas loaded with this stuff.
And when you look at how HFCS impacts this specific biological system, it is a masterclass in hacking the human body.
How so?
By combining glucose and fructose, HFCS triggers that massive, deep AgRP inhibition from the glucose, which maximizes your long-term preference and craving for that exact flavor.
Right, it turns the dial all the way down.
Exactly. Meanwhile, the fructose is driving that short-term water balloon effect. But, water passes through the gut quickly. The physical distension fades fast.
Leaving behind only the deeply altered brain chemistry that drives you to seek out that exact flavor again.
Exactly. The researchers actually showed that mice strongly prefer the HFCS blend over pure fructose alone.
Okay, wait, I have to challenge the philosophical implication of this.
Yeah.
Are you saying our deepest, most profound food cravings, like that specific brand of soda or that exact slice of pizza, are just subconscious math equations being solved by our hypothalamus?
Basically, yeah.
So we don't actually like the taste of the soda. Our brains are just addicted to the cellular drug drop it provides.
The data strongly suggests that taste is just the sensory tag your brain uses. It's a label to identify which food provides the best neurochemical payoff. The AgRP neurons are actively training your brain's future preferences based on the steepest drop in activity.
That completely changes how you look at a menu. Just think about your ultimate favorite junk food right now. Are you truly craving the flavor hitting your tongue, or is your brain simply seeking that hidden cellular drop in your hypothalamus?
It's a bit unsettling, honestly.
It is. Let's do a quick recap before we move on. So AgRP neuron activity doesn't dictate immediate meal size, but it is the master architect of future food choices.
Right. And our brains actively learn to prefer flavors associated with the steepest drop in AgRP activity.
And finally, combining glucose and fructose into things like HFCS maximizes both the preference and neural suppression, creating a highly reinforcing loop.
Perfect summary.
So we've talked about the brain and the gut. But if the volume of liquid is what's tricking the mice in the short term, how does the brain actually know the difference between the two sugars once they leave the stomach? Like, how does the gut actually communicate this complex math up to the brain?
This is where the researchers had to hunt for the messengers. They drew blood plasma from the mice 30 minutes after infusing the different sugars, just to see what hormones were floating around.
And what did they find?
A stark contrast. Fructose triggered a massive, sustained release of two gut hormones, PYY, which is peptide YY, and GLP-1. And these are released from intestinal L cells.
Okay, and what about glucose?
Glucose barely moved the needle on these hormones at that 30-minute mark.
So fructose is literally shouting its presence through PYY and GLP-1.
Exactly. But they needed to figure out which hormone was actually talking to the hunger neurons, so they essentially threw the biochemical kitchen sink at the gut.
What does that mean?
They created a cocktail blocking every known signaling receptor in the area: serotonin, amylin, CCK, GLP-1, and PYY.
Oh, wow.
And when they injected this massive blockade, fructose suddenly lost all its ability to inhibit the AgRP neurons.
Boom. So they knew one of them was the key.
Right. Then they had to isolate the culprit. They tested the blockers one by one, and only one specific blockade actually silenced the fructose, blocking the Y2 receptor.
The Y2 receptor?
Yeah, the Y2 receptor is the specific cellular docking station for the hormone PYY. When they blocked that receptor, fructose couldn't talk to the hunger neurons anymore.
But wait, what happened when they blocked that exact same Y2 receptor during a glucose infusion?
Absolutely nothing. Glucose still shut down the hunger neurons perfectly.
Oh, man. So it's almost like a dedicated subway line. Let me give you another mental image here.
Let's hear it.
The PYY hormone is the ticket. The Y2 receptor is the turnstile. And the vagus nerve is the train taking it straight to the brain. We can call it the PYY-Y2 vagus express.
That is spot on.
But just to clarify the anatomy here.
Yeah.
If I eat an apple, which is high in fructose, and a piece of white bread, which breaks down instantly into glucose, my stomach is literally making two completely different phone calls to my brain on two completely different cellular networks.
Yes. Fructose relies entirely on the hormone PYY binding to those specific Y2 receptors located on the vagus nerve. Glucose ignores that pathway entirely, relying heavily on spinal efferent nerves that run up the spinal cord.
Wow. Which raises the ultimate test. If you could temporarily mute your vagus nerve, how would your body process a sugary dessert? Let's quickly recap again before we get into how they actually tested that.
Good idea. So fructose signaling depends heavily on the gut hormone PYY.
And PYY binds specifically to Y2 receptors on the vagus nerve.
Right, and severing that vagus nerve connection stops fructose from talking to the brain, while glucose remains totally unaffected.
So how do they actually prove that final piece, muting the vagus nerve?
The researchers performed two incredibly dramatic procedures to prove this dual highway theory unequivocally.
Okay.
First, they did a subdiaphragmatic vagotomy. They physically severed the vagus nerve below the diaphragm.
Wait, they just cut the cord?
Literally. And the result was immediate. The brain became completely blind to fructose. The central hunger neurons didn't even register that fructose was in the gut.
That's insane.
But when they gave those same vagotomized mice glucose, the hunger neurons powered down perfectly because glucose was using its spinal backup network.
Okay, but physically cutting the entire vagus nerve is a pretty blunt instrument, right? I mean, it controls so many other things in the body. Heart rate, digestion.
It is, which is why they went surgical at the genetic level next. They used another viral vector, this time carrying diphtheria toxin A, or DTA.
Okay, a toxin. That sounds intense.
Yeah, they injected this into the nodoganglion.
The nodoganglion, what's that?
It's the physical cluster of nerve cell bodies for the vagus nerve sitting just outside the brain. The virus was engineered to seek out and kill only the specific neurons expressing that Y2 receptor, the PYY turnstiles.
So a microscopic sniper strike.
Exactly. The rest of the vagus nerve was left completely intact, and the result was identical. Deleting just those specific Y2 neurons completely destroyed the brain's ability to sense fructose, while the glucose signal remained strong and unaffected.
That level of precision is amazing.
It gets better. To truly visualize how precise this is, the researchers actually performed in vivo imaging of the nodose ganglion. They looked at the calcium flashes of individual single vagus nerve cells in a living animal as the different sugars hit the gut.
Oh, wow. What did the live breakdown look like?
They found that 42% of the cells responded exclusively to fructose, 34% responded exclusively to glucose, and only 24% of the cells responded to both.
It's like looking at a piano keyboard. The gut doesn't just smash its hand down on all the keys when sugar arrives. Fructose plays a highly specific, complex chord. Glucose plays a totally different chord, utilizing totally different keys.
That's exactly right.
These specific data packets are writing code directly into your brain's hardware, determining what you will crave tomorrow.
Yeah. And when we pull all of this together, it paints a really profound picture of our evolutionary history.
Right, because we didn't evolve with sodas.
Exactly. Fructose is not just a molecular twin to glucose. It engages an ancient, dedicated gut-brain highway. Think about it from an evolutionary perspective. Fructose is naturally found in seasonal fruits.
Which wouldn't always be around.
Right. It is processed passively, it promotes water retention, and it's heavily routed toward the liver for potential fat storage. It is a slow-burn energy system.
Okay, so it makes total sense that the brain wouldn't aggressively shut down hunger when sensing fructose, because historically, when fruit was available, our ancestors needed to eat as much of it as possible to store fat for the winter.
Exactly. Glucose, however, signals immediate, rapid, usable energy in the bloodstream. The brain evolved to strongly prefer the rapid neural shutdown that glucose provides because it meant instant survival energy.
Which leaves us with a critical thought to mull over as we wrap up today's deep dive.
Yeah, this is the big takeaway.
If our brains are hardwired by millions of years of evolution to chase the deep, satisfying neural drop of glucose, but fructose is processed stealthily to promote long-term energy storage without immediately turning off our hunger.
Yeah.
What happens to our biology when the modern food industry artificially marries them together? When we create high-fructose corn syrup, we are simultaneously cranking our preference dial to the absolute maximum with glucose while utilizing fructose to bypass the very neural circuits meant to tell us we've had enough.
It's a disaster.
We are feeding our brains the exact biological cheat code for overconsumption.
The science certainly points to a metabolic system that is being pushed far beyond its evolutionary design parameters. The gut and the brain are actually communicating perfectly with each other. We've just completely changed the language of the food.
So challenge yourself today. The next time you are standing in that grocery store aisle looking at a nutrition label, or, the next time you take a bite of your absolute favorite junk food, imagine those complex chords being played in your nodose ganglion. Imagine the flashbulbs going off in your hypothalamus.
Truly, pay attention to it.
Listen to your body, question your deepest cravings, and remember, your gut is always talking to your brain. The only question is, what language is it speaking? Keep exploring.